top box has a pattern of TA-TA nucleotides in the promoter region, and that helps really identify where that RNA polymerase is going to bind. So those act as an identification tool. And then once that RNA polymerase has bound, even more transcription factors will come in and attach with that RNA polymerase. Okay, so I've got my initial transcription factor recognizing that TATA box binding at the promoter. That helps encourage my RNA, that helps facilitate my RNA polymerase to attach as well to that promoter region, okay, which then signals more transcription factors to come in and attach working with that RNA polymerase. Once the process has been initiated, we then need to elongate the strand. Okay, and so the RNA polymerase is going to do the bulk of the work here. So this RNA polymerase here is going to, it's going to unwind the DNA. So it'll unwind the DNA, and it's going to base pair RNA nucleotides to the DNA. So you can see the DNA there in blue, and the RNA polymerase is unwinding it, and then it's also bringing in the messenger RNA nucleotides to complementary base pair with the DNA. Okay, RNA is made in a 5' to 3' direction, okay, which means that the DNA gene is going to run from the 3' to 5' direction. Okay, so you can see here's your RNA, that's my 5', which would make this my 3' end. Remember, things are anti-parallel. Okay, so if my 3' end is over here for my DNA, then when I follow that portion of the DNA strand, my 3' end would be on the other end of the DNA, since they're going to be anti-parallel. Remember, when the strand is elongating and we're doing our base pairing, remember RNA pairs A with U and then also C with G. So if there is a T on the DNA, because that's going to happen, I'm going to see a T on the DNA, I'm still going to put an A on the RNA. Okay, it's when I have an A on the DNA, then I'm going to put a U with my RNA. Okay, so U just takes the place of a T. One of the things we need to realize too, also with eukaryotic cells, remember with a prokaryotic cells, genes can be given, genes can be both transcribed and translated at the same time. With a eukaryotic cell, we can't do transcription and translation at the same time, but a particular gene can be transcribed by a number of transcription factors at the same time. Okay, so genes can be transcribed, multiple transcription factors can be working on the gene at any given point in time, which is important if many, many copies of that gene need to be made. Okay, so now that the initiation of the messenger RNA has started and the strand is being elongated, it's time to stop it. Okay, and so that's going to be our termination. And this is basically the RNA polymerase is just going to let go. It's going to reach a sequence that is going to trigger, in some way, shape, or form, it's going to trigger this dissociation of the RNA polymerase. So the RNA polymerase will let go and it will stop transcribing the DNA. So in eukaryotic cells, this is not very well understood. This is something, though, that in prokaryotic cells we have a decent handle on. Okay, it would help if I could spell prokaryotic. Okay, so in our prokaryotic cell, basically, there's two ways that we can have termination. There is intrinsic termination as well as extrinsic termination. So in intrinsic termination, this messenger RNA that's being formed will form a hairpin turn, basically. And it's what's called a GC-rich hairpin turn. So there is lots of guanine, lots of cytosines in this hairpin loop. And so following that loop is going to be a trail of U's, okay, lots and lots of uracils. So with my intrinsic termination, okay, I've got the messenger RNA itself forms this GC hairpin loop, Okay, that's followed by a whole bunch of U's. This causes physical stress. There's mechanical stress now pulling on the RNA polymerase. Because of this loop down here, okay, that's tugging right here on the RNA polymerase. And so this mechanical stress here is going to basically pull that U section, with a poly U section, out of the active site. And so that's going to release the RNA polymerase. Basically, it will tug the messenger RNA right out of the RNA polymerase. The other way is through what's called extrinsic termination. Okay, and so extrinsic termination, there is a protein factor called Rho, which will come in and basically just break up the interaction between the DNA template and the messenger RNA. The Rho destabilizes how the template DNA is interacting with the messenger RNA, and that's going to result in the release of the messenger RNA. So both of these are going to end up with the RNA polymerase releasing the messenger RNA so that it can now go be translated. So now that we have this strand of messenger RNA in our eukaryotic cells, it needs to be processed. Because remember, eukaryotic cells, they have those introns, the non-coding portion, okay? And this RNA needs to be modified a little bit so that it can be translated. So one of the things that will happen is they will add what's called a 5' cap. Okay, that 5' cap, okay, has a bunch of, essentially it's a modified guanine molecule. Okay, so our 5' cap, okay, which is a modified guanine molecule, is added to the 5' end. Okay, you can see the cap there expanded out so you can see what it is. Okay, on the 3' end, we get what's called a poly A tail. Okay, and so that poly A tail is going to consist of about 30 to 200 adenine nucleotides. So that's on my 3' A, basically just a whole bunch of adenine nucleotides added on there. And the purpose of these two things, okay, they do a variety of functions. The first one is they help prevent degradation. As that RNA moves out into the cytoplasm, it helps prevent the cytoplasm basically from breaking down the messenger RNA. I'm sorry, it's not a protein yet. Okay, so it helps prevent degradation. Okay, they act as signals to help the ribosome, so that the ribosome knows where to bind to the messenger RNA. Okay, and then the poly A tail, okay, it also is going to determine how many times that messenger RNA can be translated before it is going to be destroyed. Okay, so they prevent degradation, they help the ribosomes figure out where to attach to the messenger RNA, and the poly A tail is going to determine how many times that messenger RNA can be translated by ribosomes before it is too degraded to continue on. In addition to the adding of the 5' cap in the 3', the poly A tail on the 3' end, the introns also need to be removed from eukaryotic RNA. Remember, our introns, these are our non-coding regions. So you can see in my pre-RNA here, I've got this intron that is not going to code for anything. It's what's considered junk DNA. And so that needs to basically be just cut out. And it's cut out by what's called a spliceosome. Okay, so the spliceosomes are going to remove the intron. Spliceosomes are going to be made of SNRP, okay, what are called SNRPs. Okay, so SNRPs and proteins will combine to form the spliceosome. If you remember some of the different kinds of proteins we talked about at the very beginning, there's an snRNA. Okay, and so that spliceosome is going to go in and splice or cut the DNA. Okay, and so it will cut the intron out, and it will hook the exons then back together. And in some types of RNA, those exons can be recombined in different ways. For example, with the one on the left here, okay, once this intron is cut out, those the exon on the left and the exon on the right are just joined together. Okay, but as you can see over here, okay, you can recombine how these are spliced together. Okay, we can do different combinations of exons. Essentially what you have there is, in this case on the left here, the green was our intron, and on the other case, the yellow was the intron. Okay, but those different exons can be combined in different ways, and that can lead to a different number of proteins, a different kind of protein, okay, which can be very important for from an evolutionary standpoint. So once we've finished all of this modification, we've taken, again, especially in a eukaryotic cell, we've taken this pre-messenger RNA and we have turned it into mature RNA, mature messenger RNA. And this mature messenger RNA is now actually ready to be translated. So now that we have this RNA strand, this mature RNA strand, we need to take it and turn it into a protein. And that's going to be the translation portion. Okay, when we take that messenger RNA strand and we turn it into the protein, the polypeptide chain. Remember, that's going to occur out in the cytoplasm at the ribosome. So let's take a second and look at the structure of the actual ribosome. So the ribosome is made up of two parts, what's called the large subunit and the small subunit. And those two parts are separate from one another unless translation is actually happening. Okay, the initiation portion of translation actually brings those two portions of the ribosome together. The ribosome itself is made up of proteins as well as that rRNA that we talked about before that helps to facilitate the connection between the messenger RNA and the transfer RNA. Several ribosomes at once can translate the same piece of messenger RNA, but they will all have the same basic structure, the large and the small subunit and these three attachment sites. And so the three attachment sites, we've got our exit site, our what's called the P site, and the A site. Okay, the A site, this is arrival. Okay, so this is where the tRNA arrives. Okay, and it's going to arrive with its amino acid, and it will basically land in this A site, this first attachment site. Okay, the P site is where the tRNA that's attaching these two, both these. So you can see in this third picture down here, you've got a tRNA in the A site and a tRNA in the P site. And the one in the P site is going to attach to the one that's there in the A site, adding its amino acid to the growing polypeptide chain. And then the E site here is going to be the exit. So now this transfer RNA that no longer has an amino acid is going to exit from the ribosome. So the transfer RNA itself has some particular characteristics to it. Okay, so the transfer RNA is what's going to be bringing the amino acid over to the messenger RNA. So it's got a site for attachment of the amino acid to it. Okay, it forms a kind of a clover shape here because of the complementary base pairing. You can see it's self-base pairing here. Okay, which bends it into that clover shape. And then on one end, it has the anticodon. The anticodon is going to be able to base pair with the codon on the messenger RNA. Remember, the messenger RNA is grouped into threes, okay, and those three are, just making stuff up right now. Okay, those groups of threes, remember, on the messenger RNA are called codons. And so the transfer RNA has the opposite anticodon on it. For instance, on this transfer RNA here, the anticodon is AAG, which means the messenger RNA would read UUC. Okay, and by pairing this, that's what allows it to know what amino acid it needs to bring over. Something you do need to be aware of in a transfer RNA is what's called the wobble position. Okay, the base pairing rules in transfer RNA are not as strict as they are in other places. Okay, so especially in that third position, and that's because of that code redundancy. Remember, we have a lot of amino acids that have more than one code for the amino acid. So that allows for some flexibility in that third base pairing position there. Okay, and so that third base pairing position there is called the wobble position. For instance, a U on an anticodon, okay, combined with an A or a G on a codon. Okay, and so that's that wobble position, which shouldn't change the amino acid that should be there. So we're going to have to get these amino acids actually attached to the transfer RNAs. And they're going to